Thank you for joining me for this extended overview of FlowLogic 7, a flow cytometry analysis package that combines gating, graphing, statistical analysis, and reporting tools. I will start by covering a few of the important aspects of the program preferences before moving on to the analysis basics, including importing files, creating and managing gates. I will then demonstrate a complete analysis workflow utilizing the three main sections of FlowLogic, being GateLogic, GraphLogic and DocLogic, before moving on to some of the more advanced features, including PlateLogic, which can be used to generate heat maps and screen plate data and other large data sets, Titrate Logic, an automated titration analysis tool, the new global analysis templates, which can include everything from gates right through to reports. I will demonstrate cell cycle and proliferation analysis, auto-compensation, and Boolean gating. This FlowLogic update includes many new features, the main ones being a redesigned GraphLogic with a range of new graphs and an upgraded spreadsheet, a completely redesigned DocLogic with a panel of new formatting and editing tools, global analysis templates, which can include everything from gates, heat maps, overlays, graphs, and reports, the ability to merge or combine analysis files, and a new instant Boolean Gate generator. There is also a dramatic increase in processing speed and an increased capacity to handle extremely large files and very large datasets. I'd like to make two more points before I begin. The first is that FlowLogic software is multi-platform compatible, as it is the same piece of software that operates on Windows, Mac and Linux computers. And this means that an analysis can be performed, for example, on a Windows computer, saved and reopened on a Mac. And on that point, there are two different analysis file types that can be created. The first is a .glf, which stands for GateLogic file, and this saves the analysis and creates a link to the FCS or MQD files. The second is a .GateLogic experiment file, and this compresses all of the FCS files and the analysis into a single file. And this is very useful if you plan to move or share the analysis. So here we have FlowLogic, but before beginning your analysis, it's worthwhile reviewing some key aspects of the program preferences. In Apple, the preferences are found under the FlowLogic menu, and in Windows, under the Edit menu. The preferences are divided into different categories down the left-hand side, beginning with the General Preferences, which contains a range of different plot display options. It's also recommended to enable the auto-saving at a defined time interval. Under Parameters, the user can decide whether to display the user labels created at the time of acquisition or custom labels that can be changed in FlowLogic. Under Export, the default elements to be included with the plot image can be set, along with the resolution. Under User Interface, you can choose to display FlowLogic in a single window view, as I am here, or as a split window view if you have external monitors. And to maximize speed and performance, under System, set the allocated memory to that which is available in your computer. I will now move on to the FlowLogic analysis basics, being importing files and drawing gates on plots. Imported files appear here in the file inspector, and the easiest way is to drag and drop files or folders containing files here into the file navigator. And as you can see, my new experiment folder containing my files has taken its name automatically from the imported folder. And my imported files appear here in the file inspector. Right-clicking on a file opens up a menu allowing a range of different renaming options to be applied to individual files or all files in the folder. Samples can be displayed as dot plots in the workspace through the right-click menu or simply by double-clicking on the file name. Above the workspace is a toolbar with the various gating options. And for example, to create a polygon gate, we select and click to define the region. Gates can be named when they are created, or later by right-clicking on the gate name in the file inspector. Polygon gates can be adjusted by clicking and dragging a corner, clicking and dragging an edge, or moved by clicking and dragging from the middle. The daughter population can be opened by double-clicking on the gate in the file inspector or from within the gate itself on the plot. To change parameters, simply click on the parameter name and choose a new one from the list. In addition to polygon gates, there are rectangle and elliptical gates, histogram markers, and, as I'll demonstrate here, quadrant gates. Once created, the quadrant gates can be adjusted by clicking and dragging the center or individual arms. And gates can be deleted through the right-click menu in the file inspector or by highlighting and clicking the trash can icon in the toolbar. Gated populations can also be colored, and I'll demonstrate this with the rectangle gate here through the plot side draw. As you can see, there are a range of tabs, and here in the gate list tab we can turn gates on and off without having to delete them. Choose a color and tick on to color the gated population at every level of the hierarchy. And as I move this gate, the colored events and the gate statistics update in real time. The colored events are also tracked when I change parameters, and we can color multiple populations across different parameter combinations. A handy tip is that you can return to the parameter combinations where a gate was created simply by clicking on the gate name in the plot side draw. In addition to coloring gated populations, the plot display can be changed through the different options in the toolbar. For example, dot plots, histograms, contour plots, contours with dots, pseudo color plots, and a range of combinations of these displays. Once gates have been created, they can be copied from one sample and pasted to other samples using keyboard shortcuts. Drag and drop can be used to copy gates from one sample to another, or to copy gates from one level of the hierarchy to another, even within the same sample. Selected gates can be copied using the option in the right-click menu, and then pasted either to all samples, or to a selection of samples. To copy a gate to a different level in the hierarchy, select the gate, right-click, choose Copy Gates, then select the level, right-click, and choose Paste and Insert Gates. Gates can be deleted, as shown before, by highlighting and choosing the trash can from the toolbar, or by selecting the gates in the file inspector, right-clicking, choosing Rows, and Delete. A window will then ask to confirm this action. Gating templates can also be saved by selecting the file, right-clicking and choosing Gate and Save Gate Template. And this can then be applied to samples within the folder by right-clicking, choosing Template and Apply Gate Template, or to samples in future analyses. It's also useful to know how to handle the workspace. For example, if I open a range of plots, I can resize all of them at once using the slide bar in the File Navigator. However, it may be more useful to define the number of plots to be displayed horizontally, and this is found under the Edit menu and Plot Arrangement. And here, if I set this to 4, I can display one sample per row. The workspace in FlowLogic is dynamic, meaning that individual plots can be resized and all other plots will move around it accordingly. Plots can also be freely moved around the workspace, but at any time can be reordered using the slide bar, with the order of the plots in the workspace matching the order in the file inspector. Now that we've covered the basics, I'd like to demonstrate a complete analysis workflow utilizing the three components of FlowLogic, being Gate Logic, where gates are created, Graph Logic, where results can be graphed and statistical analyses can be performed, and DocLogic, where reports can be generated. One of the best features of FlowLogic is that these three sections are all fully linked, so after adjusting a gate in Gate Logic, all statistics, graphs, and reports are updated automatically. One feature that makes this auto-update even more effective is group analysis. Once files are placed in the group, any action performed on one sample is automatically applied to all other samples in the group. These include creating and adjusting gates, calculating statistics, and changing the plot display. To group a set of files, select them in the File Inspector, right-click, choose Group, and add to New. The new group is then defined by a colour, in this case green. Now you can see that as I change a parameter on one plot, all other plots are updated. And likewise, when I draw a gate on one plot, the same gate is instantly applied to all other samples. The same also applies to the naming of gates. Other useful little features include the ability to close all plots at once, and also the ability to select a particular level in the hierarchy for all files. And this can be useful for opening a selection of plots, deleting certain gates, and also exporting a selection of plots. Using this method of closing all plots and then opening all plots from a certain level of the hierarchy, you can see I can analyse my 15 files and visually assess the gate on every single sample. Selected populations can also be removed from the group by right-clicking, choosing Group, and Remove. And you can see this first rectangle gate can now be moved independently. Additional groups can also be created for a selection of gates, as you can see here. And groups can exist across experiment and project folders. And finally, selected gates can be added back to an existing group. Another method for viewing all samples whilst gating is to create a plate in PlateLogic. This can be done by right-clicking on the experiment folder and choosing to build a plate. And it will build a plate with one column for each sample. Now I can work on one sample at a time, and you will see as I draw a gate, the plate will be updated with a new row for the resulting daughter population. I can now continue my analysis on this one sample by creating two more gates. This is the perfect way to assess every single plot whilst analysing, and the plate itself can be exported or added to a report in DocLogic. Now that I have finished gating my samples, it is time to start investigating the populations in more detail. And the first thing I want to do is perform some live backgating. To do this, we can open the plot side drawer by clicking this button, choosing a colour and ticking on. And this colours the events at all levels of the hierarchy, and in this case, for all samples in the group. Again, we can colour multiple populations, and using group analysis in combination with the plate view, we can view our coloured populations on every single plot in the entire analysis all at once. Statistics can also be viewed in the Advanced Functions drawer, and to do this, in the Set Statistics tab, tick the relevant gate or parameter statistics and view the results in the View Statistics tab. Right-clicking on the table allows you to copy or export the statistics. It is also very easy to create overlays in FlowLogic, and the fastest way is to select the relevant populations from the File Inspector, right-click, and from the menu, select Overlay and New Overlay. And by default, this adds all samples to a dot plot overlay. And this plot acts like any other plot in that you can view it as a histogram or click on the parameter labels to change the parameter. Right-clicking on the overlay brings up other options such as colouring the histograms or splitting the histograms. Now that the gating has been performed, the next step in the workflow is to graph the results and perform statistical analyses using the features in DocLogic. In terms of appearance, DocLogic resembles GateLogic in that the files are listed in the File Inspector on the left-hand side. Graphs are displayed in the graph window at the top of the screen in a similar way to the display of dot plots in GateLogic. And instead of the Advanced Functions drawer, there is the Workspace spreadsheet. And this is where all forms of data can be imported and rearranged. It is worth noting that the gating hierarchy is hidden from the File Inspector, as the population selection is performed in the workspace, as I will demonstrate. So to import our FCS link data, we select our samples in their experimental groups and drag them into the workspace. This generates a graph table with a table name, group names, and sample numbers. Now when I click on a data point in the table, I can view or change the gate, the statistic, and the parameter, if relevant, for that sample. Or I can select multiple samples and choose a new gate and change the statistic. I can also update the group names, change the table name, usually to match the statistic being displayed, and I can use the other options in the toolbar to format the workspace in terms of the cell or text color, the font style, type or size, the number of decimal places to be displayed, and a range of values can be defined as being technical replicates. So here I am spending a little bit of time setting up my table, but once it is done for one population, it is just a matter of copying, pasting, and then defining a new statistic or population. And that is exactly what I am doing here. I am pasting the original table 2 times, changing the table name to a different population in this case, and then it's just a matter of selecting the data in the second and third tables, and I will choose a new population. So once I have my data all set up in the tables, it is very simple to graph the results and perform statistical analyses on the data. To do this, I highlight the relevant data, then click and drag it into the table folder in the folder system to the left of the workspace. Then double-click or right-click and open on the Graph tab to display the graph, and to perform statistical tests, select the Analysis tab, choose the desired test and the groups to be compared, and the result will be displayed to the right. The remaining data can be graphed very quickly as well by right-clicking on the workspace folder and adding additional table folders, selecting the new dataset and dragging it into the new table folder. And you will see that the table folder name is automatically applied from the table name in the workspace, these being the cells coloured yellow in this case. So as you can see, I was able to graph these 3 populations very quickly, and now that I have my table set up, I can again copy and paste and choose a different statistic. For example, the event count, and as I'm changing the whole table, I can change it all at once. I can also import non-FCS linked data by right-clicking in the workspace and choosing the import option from the menu. The entire workspace can also be exported by right-clicking and choosing Export Table, and this can be saved as a CSV or an XLSX file. I would like to emphasize that one of the huge benefits of FlowLogic is that these 3 sections, GateLogic, GraphLogic, and DocLogic, which we will see soon, are all fully linked. So if I was to move a gate in GateLogic, all of the statistics here in GraphLogic and the graphs would all be updated automatically. Now that we have graphed our results and performed some statistical tests, I'd like to spend a bit of time describing the new features in this version of GraphLogic. One of the big improvements is the way that graphs are formatted. You can see when a graph is selected, different labels are displayed in the corners of the graph window, and clicking on any of these opens up the corresponding formatting panel on the right hand side of the screen. For example, click to change the graph type, show or view stars and p-values, display different statistics and error bars, select the legend formatting options, or click the info button to get a description of the different areas that you can click on in the graph. You can also click on different components, such as the bars here, which gives you options to change the color. You can also click on the different axes and axis labels. And there are different options within these specific to that part of the graph. For example, we can add 1 decimal place to the y-axis values. And here we can rotate the labels on the x-axis. You can see that you can also click and drag different components, such as the legend or different titles. You can also edit the title and change the font size, type, and style. As I will demonstrate shortly, any changes made to graphs here in DocLogic will be maintained when they are imported into a report in DocLogic. Additionally, in DocLogic, there is also the option to copy and paste settings from one graph to others. So now, if we move to DocLogic, on the left-hand side, we can navigate to our list of GraphLogic tables, statistical analysis summaries, and graphs. Select and drag in the graph that we were working on. You can see that it has maintained all of the formatting changes. You can also see that we can continue to edit this graph by clicking on the same areas, and we see the same formatting panel appear on the right-hand side of the DocLogic window. And here I can quickly change this graph from a bar graph to a box and whisker plot. DocLogic itself has also been redeveloped, with much of the additional functionality being available from the icons in the toolbar, which I'll demonstrate in a moment. You can also see the 3 tabs at the top of the file inspector, and this is where we can import dot plots, overlays, plates and plate components, and our graphs and associated tables. Now if I import a dot plot onto the page, you can see by clicking on it brings up a corresponding analysis window specific for this element. For example, here I can lock the aspect ratio to change its size proportionally, and this can be done using the up and down arrows, clicking and dragging a highlighted corner, or typing in new height and width values. You can see that you can also click on the plot title to change the font, style, type, and size. I will now demonstrate the distributing, aligning, grouping, and reordering options available in the toolbar. To do this, I will import 4 plots onto my page, select them all, and then click on the corresponding icon up here in the toolbar. So first of all, I will click to align by the top of the plots and distribute horizontally. By selecting them, I can choose to group them all, and now they are one element on the page. Likewise, I can select the grouped element and ungroup them. If I resize one plot, I can choose to copy the plot setting and then paste the setting either to a selection of other plots or for a selection of reports. So here I will paste the size setting to these other 3 plots. We can now also copy, cut, paste, and clone a selection of items. For example, to clone this plot, I select it and click on the icon in the toolbar, and a copy is automatically generated. Here you can see I can copy a selection of plots, add a new page to my report, and paste to paste them into the same position on the second page. Other icons in the toolbar allow you to insert a text box, table, import an image, add a keyword text box, add lines, arrows, and shapes. So here I will add a rectangle, and once again you can see if I click on the rectangle, the corresponding formatting window appears. So here we can fill the rectangle with a color, change the border, and also lock the aspect ratio. If I create a clone of this rectangle, change the color, I can demonstrate the reordering options available in the toolbar. These allow objects to be sent to the back, brought to the front, or stepped back or forward one level at a time. Now if I add a text box to the page, you can once again see that each element has its own unique formatting panel. So here, if we type in some new text, with the formatting panel on the right, we can change the font size, type, style, and color. And using the copy and paste options, you can have the same title text box, for example, on each page of a report. This method can also be used to generate headers and footers, or to create a text box as part of a template that you can fill after each analysis. Lines and arrows can also be added with the click and drag used to define their length. A new feature to DocLogic is the ability to add a keyword text box, and this allows metadata associated with individual files to be added to a report. So, draw the keyword text box and then click and drag a single file into the text box. You will then see a complete list of the metadata keywords and their values. Select the relevant values and then click and drag them into the text box. And you can see the summary here. This is another feature that is very useful when generating templates with the particular file that populates the keyword text box, determined by its order or rank in the file inspector list. Empty tables can be added directly to a report using the icon in the toolbar, which also allows you to define the dimensions of the table. Once on the page, double-click to add text to the cells. This type of table is independent as it is not linked to any data. However, data tables linked to the workspace in DocLogic do remain linked to the files that were used to create them, and so adjusting a gate would in turn update the values in the table in the DocLogic report. So here, if I import a graph and the linked table folder data, you can see it takes the formatting as was defined in DocLogic. If you click on the table and then deselect the original settings option, this allows you to format the look and the colors of the table in the report. However, the numbers remain linked to the original data. And here I can demonstrate how only the selected text will be formatted using the options in the formatting panel. In a similar way, the statistical analysis summary tables can be formatted in terms of their color and text style, but the data itself remains linked to the original files. As a result, if a gate is adjusted, then the values in the table will also update. And finally, plates from PlateLogic and all the associated graphs, tables and summaries can be added to the report via the Plate tab at the top of the file inspector. And plates themselves can be formatted in terms of their font and also their size. The final icons I'd like to highlight are those that exist at the top of the page preview and the list of reports. The page preview can be viewed by clicking on any empty part of the page, and here are options to add a new page, clone a page, insert a page, export a page and delete, and similar options exist at the top of the list of reports. And documents can be renamed by double-clicking on the title. I'd now like to briefly demonstrate the copying and pasting of graph settings. So here, using the icons in the toolbar, I will copy the settings from the first graph and paste them to the others. And you can see that everything is transferred except for the titles and the group colors. You can also choose to paste the settings to entire documents. The most powerful new feature in FlowLogic 7.3 are the global analysis templates. These templates can contain any element from GateLogic, DocLogic and DocLogic, and so applying a template to an experimental folder can automatically apply the gates and generate reports. So here I'll demonstrate applying 3 different templates to 4 different folders. The first template will be applied to 2 folders with datasets representing 2 different time points. And this template includes overlays, a plate view of the hierarchy, graphs, and a 2-page report. Once the template has been applied, the analysis is just like any other analysis, in that gates can be adjusted, groups can be changed, and you can add or subtract different elements. So here I can click on the 2 different folders and you can see my 2 different data sets. I can add my second data set to a new group if I want to adjust the gates on this data set by themselves. And here in DocLogic, you can see 2 new reports have been created, both with 2 pages each. And you can see As I switch between the second pages of both reports, we have completely different results. This second template is being applied to 96 well plate data and includes a heat map and two separate reports. There are in fact two plates saved in this template, with the second one showing the histograms coloured to match the heat map. And as mentioned, here in DocLogic, there are two separate reports generated for this one experiment folder. And this third template includes the setup for a cytokine bead array. And it includes all information such as the analyte names and the expected concentrations for all of these standards. So once it has loaded in PlateLogic, it is ready to be sent for further analysis in bead logic. Now I'll give an overview about how to set up an analysis in order to save the template from the experiment folder. So the important factor is that you only have one experiment folder in the analysis. You can see in GraphLogic you can have multiple workspaces and multiple table folders within each workspace. And in DocLogic you can have one or more reports. But the important thing is to have the single experiment folder in GateLogic. So once you are ready to save the template, right-click on the experiment folder and choose Save Template from a folder. And likewise, select Apply Template to Folders when you want to apply a template to one or more selected folders. It's important to understand that the folder that you are applying the new Global Analysis template to must have the same number of files as did the original folder that you saved the template from. This is because if there are too few or too many files in the folder, FlowLogic won't know how to handle the extra files. So it is worth considering when you are building a template to save it with the maximum number of files that you think you will be analysing. It is always easier to delete the files and linked components once the template has been applied. It is also possible to name documents using keywords in the file metadata. This may be useful if you'd like the file name from the first file in the list to be what names you document as part of your template. To do this, select the document, then select the file containing the metadata and drag it on top. Then from the list of metadata, choose the keyword or keywords that you'd like as part of the title, then click and drag onto the document name. In addition to global analysis templates, this version of FlowLogic for the first time allows the importing of saved analysis files. To demonstrate this feature, I will first of all open a saved file by choosing File and Open and selecting my saved GateLogic experiment file. And you can see it loads here with 15 files. This contains a report with 2 pages. And now if I want to import a different saved file, I choose File and then Open Append. And this will import that saved analysis, creating a second project with its associated experiment folder. However, this can be moved and reordered just like any other analysis. So here I have my 2 different experiment folders with analyses performed at different times. And you can see these now both exist in DocLogic and GraphLogic. One of the huge advantages of being able to import an entire analysis is that those newly imported files can then be analysed directly with the existing files. So here I will perform a grouped analysis where I will combine the files from 2 different time points. To consider the 2 independent variables, I will paste the tables for each time point side by side. I will then colour these just to help distinguish them. Then the next step is to merge the cells directly above the column count. To do this, highlight the cells and click the Merge button in the icon. You can now add the label for the second independent variable, in this case the 6-hour and 24-hour time points. And now it is simply a matter of right-clicking on the workspace, adding a new table folder, highlighting all of the data and dragging it into the folder. And as you can see on the graph, we have our two different conditions and our two different time points. The two-way ANOVA is also new to this version of FlowLogic, and when combined with a Bonferroni multiple comparisons post-test, we can automatically add stars of significance and p-values to the graph. So under the Analysis tab, if you choose the appropriate test, you will see in the final column, where we have performed the multiple comparisons, you can choose to show the stars of significance. Then, in the stars formatting panel, you can display and format the stars, connecting bars, and p-values. And the stars and p-values displayed on the graph will update as any gate is adjusted. So PlateLogic is a tool for the screening, filtering, and visualization of large datasets, such as plate data, and it contains a range of features including automatic heat map generation and outlier detection. So here I will use PlateLogic to perform a data array analysis, and I will use tags to help filter the results. Firstly, to help identify our tags, I will move the tags column right up next to the file name. In their simplest form, user-defined tags can be added to one or a selection of files by highlighting, right-clicking, choosing tag, and adding new tag. And for example, we could call this Group A. Likewise, I can select a new set of files, right-click, choose tag, add new tag, and this could be Group B. Now, if we right-click in the file inspector, choose tag, filter, and Group A, we can display just those samples in Group A. Now, if we right-click, choose tag, and set our filter mode to any tag, and then select Group B, we can display only the files that are in Group A or Group B. It is also very easy to clear all filters. FlowLogic also has a range of more advanced auto tags, which can be particularly useful when performing data array analysis with heat maps. To produce a heat map, select PlateLogic from the Advanced Functions pull-up drawer, click in the Plate window and select New Plate. Then, highlight all of your files and drag them into the plate, and you will see PlateLogic automatically generates a heat map. To help analyze all of these files, I will also place them in a group. Opening the Plate side drawer reveals a number of tabs, the first being the Plate Setup tab, the rest being the heat map settings, statistics, plot viewing, advanced functions, and outlier detection. Double-clicking on a well will open the corresponding sample in the workspace, and this can then be analysed just as you would any other sample. So here, I will arbitrarily draw a gate on these two parameters, and as this file is in a group, this gate will automatically be applied to all other samples in the group. Back in the Plate side drawer, I will change the well shape, and in the Heat Map Settings tab, I will choose this new gate and change the statistic to percentage of parent, and you will see a new heat map is automatically generated as the automatic heat map setting is on. Of course, the user can customize the heat map by turning off the automatic setting, double-clicking in the value column and typing in a new value. So as you can see, this could potentially be a very useful way for identifying samples that sit above a particular threshold. These results can be displayed under the Statistics tab in a table that can be coloured to match the heat map, and this colouring will be exported with the tables if they are displayed. These results can also be displayed in columns, which again can be coloured to match the heat map. Comments can also be added to individual samples. In the Plot Viewer tab, individual dot plots or histograms can be viewed simply by hovering over the wells, but the plate itself can be set up to display plots rather than the heat map colours alone. And this is done by right-clicking in the plate and choosing Show Plots. And this can be in the form of dot plots or histograms. And these can also be coloured to match the heat map colours. Where auto tags can become useful is when we only want to display particular samples that sit in a particular range in the heat map. To apply the auto tags, right-click anywhere in the file inspector, choose Tag, Auto Tag, and in this case, Plate Heat Map. And you will see the corresponding heat map color has been assigned to every sample in the list. Now we can apply a filter to display only those samples that sit within a particular range in the heat map, in this case, those colored red. And here we have those 20 files that sit within this range. These files can now, for example, be selected, moved to a different folder, and analyzed independently. Now I would like to demonstrate Titrate Logic, which is our automated titration analysis tool. Here I have my CD4 titration files, to which I will apply a previously made gating template. I will now group my files, identify the population being titrated, and display that parameter on the x-axis. Then I right-click and click Titrate Logic. FlowLogic then creates histograms for that parameter and automatically detects the positive and negative peaks. It will also create 2 new titration overlays and display all of the plots in a plate. In DocLogic, a report has been automatically generated showing all of the plots, the 2 titration overlays, and graphs showing the positive and negative MFI, the signal to noise ratio, and the stain index, with the highest point on the stain index graph representing the optimal dilution. A text box is also created where important information can be recorded, such as the antibody clone and lot number. I would now like to quickly demonstrate cell cycle and proliferation analysis. For cell cycle analysis, display your DNA content parameter as a histogram, open the side drawer, click the Cell Cycle tab, and from the drop-down menu, choose one of 5 algorithms. The area under the curve, representing the different stages of the cell cycle, will then be displayed in the plot side drawer. Proliferation analysis works in a similar way. Display your data as a histogram, and then in the plot side drawer, choose the Proliferation tab. Define the position of the background and the loss factor, and then click Fit. And FlowLogic will match the curves to each peak. And the results can be adjusted by changing the position and width of the gate for the first generation. Autocompensation can be performed given the appropriate single color controls. To perform autocompensation, select CompLogic from the Advanced Functions pull-up drawer. Then, click on a file in the File Inspector to load all of the files in that experiment folder in the FCS Files window. From the list of loaded FCS files, select the single color controls and click Set FCS. From the parameter list, choose the parameters that you wish to compensate, and then click Set Parameters. The next step is to match each compensation file with the parameter, and you can move the columns to make this comparison easier. If these don't match, click on the control file name and choose a new control from the drop-down list. Once the parameters and control file names have been matched, click Set Gates. FlowLogic will then draw a polygon gate on the forward versus side scatter plot for each sample, and create positive and negative histogram markers on the resulting daughter population. Both the polygon gate and the histogram markers are examples of auto gates, with the polygon gate automatically detecting the densest population on the forward versus side scatter plot, and the histogram markers automatically detecting the positive and negative histogram peaks. If required, you can adjust the polygon gate, and as you can see in the file inspector, the polygon gates have been grouped, so adjusting a polygon gate for one sample will update the polygon gate for all samples. Likewise, the histogram markers can also be adjusted so that they accurately fit the positive and negative populations. Once the setting of the histogram markers is complete, return to the Advanced Functions pull-up drawer and click Compensate. FlowLogic then creates clones of each of the polygon-gated populations and displays each fluorochrome against every other fluorochrome. A line is then created on each plot based on the median fluorescence of the positive and negative populations, with a perfectly horizontal line indicating perfect compensation. If the line is not perfectly horizontal, the compensation can be adjusted manually in the plot side drawer under the Interactive Compensation tab. The median fluorescence values for the positive and negative population for the parameters being displayed are shown in the Interactive Compensation window, and the values for all samples can be reviewed in the Statistics window in the Advanced Functions pull-up drawer. Additional display tools allow you to show the full display for all plots, which will allow you to see all events, including those sitting below 0 on the axis, and also to switch between log-scaled and traditional logarithmic scaling of the axes. When reviewing the median fluorescence values in the Statistics window, clicking on a plot in the workspace will highlight the corresponding row in the Statistics window, and vice versa. Once the compensation has been confirmed visually, the new matrix can be saved and applied to files at a later date, or it can be applied directly to all files in a particular experiment folder. The matrix and all dot plots resulting from the autocompensation can be sent to a report, and at any stage click Finish to clear the process and start again. The Boolean Gate features can be found under the Boolean Gate tab in the plot side drawer. Here, the Boolean Gate expressions can be created using the drop-down menu to select the input gates and the commands listed in the toolbar. In this example, I will create an inverse gate or a NOT gate. To create my inverse gate, I select NOT, choose the population, and click Add. Here, I can also choose a color for the new population. And now you can see all events outside the gate have been colored, and these events are colored at all levels of the hierarchy, as you would see with any normal gate. And likewise, I can rename this population in the file inspector and open it as a dot plot. And you can see these plots update in real time as the original gate is moved. A new feature in this version of FlowLogic is the Auto Boolean Generator. So, in the plot side drawer, under the Boolean tab, click the Auto button, and this will open a window with all of the possible Boolean combinations. Ticking the box next to the different Boolean expressions will instantly generate that gate. So I hope you have found this overview useful. We will be creating a range of more in-depth videos in the near future.